Now that we've made NADPH and we've made ATP, we can move on to the Calvin cycle, where these two molecules will be put to work. The Calvin cycle takes place in the stroma, the thick, gooey, chemical-rich core of the chloroplast. And this is going to look like a very complicated cycle, but we are going to learn a few parts of it and not the entire thing. Okay? So first of all, in all reality, 3 carbon dioxides come into this system at a time, 1 per cycle, but we're going to kind of follow all 3 at a time here to see how this works. Okay? So 3 carbons come in. And they come in through the stomata, they enter the leaf, they go into a mesophyll cell and make their way into the chloroplast. Now, the first step of this is phase 1, carbon fixation. Carbon fixation is when carbon is taken from a gas and turned into a physical solid. Okay, so what happens here first is there's an enzyme that I'd like you to know right here called rubisco. Rubisco, it's kind of easy to remember. It's the most common enzyme on the planet, I think. So there it is, rubisco. And CO2 comes in, it sticks to the active site on rubisco, and it reacts with RUBP, which is the last product of the last round of this Calvin cycle. So rubisco reacts CO2 and RUBP. Okay, we're not going to worry about phosphoglycerate. There we go. I don't know why I couldn't say that. We're not going to worry about phosphoglycerate, the intermediate, but the first reactants are CO2 and RUBP catalyzed by the enzyme rubisco. And this is called carbon fixation. Okay, the next phase is called reduction. You know what that means. That means that electrons are added. So some of the ATP we've made is put to work here. Some of those electrons that we've sent this way are utilized, right? They're donated to this. And the final product, or the product at this point, at the end of the reduction phase, is G3P. G3P is kind of like pyruvate, and there's a phosphate group stuck to the end of it. Okay, it's kind of like pyruvate, and there are 6 of them made at the end of reduction. There are 6 of those things made. One of them leaves. And basically what G3P is 1/2 of a glucose. And you get 1 of them here. But that's where these 3 carbons go, right? They end up right here in these 3 carbons in G3P. But there's now 5 G3P left. So what do we do with those? Don't we get more glucose out of those? No, we do not. They are now regenerated. So phase 3, regeneration. In regeneration, we take 5 G3Ps, which you should know that one, and they are converted with the help of some more ATP, converted to RUBP, which is the final product of the Calvin cycle, as well as the first reactant that's going to be fixed to CO2 in the next round. Okay, so we can see here that to make 1 G3P, we've had to utilize 9 ATP, and we've had to utilize, let's look, 6 NADPHs. So we need more ATP than NADPH, which is why we had cyclic electron flow earlier, right? And now Now let's do some more math. If I asked you this on a test, how many NADPHs and how many ATPs do you need to make a glucose? Well, you know that you need 9 ATP to make a G3P, which is a 3-carbon molecule. How much would you need to make glucose, which is a 6-carbon molecule? So for one glucose, you would need 18 ATP, and you would need 12 NADPHs, right? That's what you would need to make a whole glucose molecule that has 6 carbons in it. So this is a cycle because the last product is also the first reactant, and every time you bring in 3 carbon dioxides up here, that's the input, 3 CO2s, you're going to spit out a G3P, a half of a glucose molecule. Now that might not make glucose, it could make fructose, it could make other things, but we use glucose as our example. So G3P is that 3-carbon molecule. A lot of times it's converted to sucrose or table sugar for transport by the plant, and then later on it's turned into starch for storage. Why starch? Because starch, as we'll learn later, is I'm just gonna draw a box to represent glucose. Starch is a polymer of a bunch of glucoses stuck together, and therefore it's big, so it can't drift out of the cell by accident. You can store it, and it's not soluble. It does not dissolve, and that is important because it doesn't affect osmosis, causing the cell to swell up with too much excess water. So it's not soluble. It's big. It doesn't accidentally get dissolved or it doesn't float out of the cell by accident, and so it's a good storage molecule. Now when the plant wants to, it can break this starch down into an individual glucose and then run that through the process of cellular respiration, and then it will get about 30 ATP back out of that glucose, just like an animal would. So they do the same process as we do as humans. Now you can see then that the light reactions take place in the thylakoid. You've seen all this already. This is all of this tied together. So we have an organism here. This organism has organs, that has cells. Inside of those cells are organelles, where all of these chemical reactions take place. Sugars are produced this way. Sucrose, which is basically a glucose stuck to a fructose. And that's where the foundation of our food web is, where producers are eaten by herbivores, predators eat herbivores, so on and so forth. And at the same time, this is the source of all of the oxygen that we breathe on the planet. All the oxygen we breathe on this planet was produced by living things. There was not really much oxygen at all on our planet originally. All of it is basically the poop of photosynthetic organisms, and they modified the planet. They changed the planet and made life possible for the life that we have today. One other thing I'd like to mention are some modifications to photosynthesis. Sometimes when it's really hot, plants will close their stomata. So there's openings in the leaves. Remember, those openings let carbon dioxide in and they let oxygen out, right? And when it's really hot, they close them up. Why? Because they also dehydrate out of those openings. And when that happens, there's a problem. When they shut and they have a whole bunch of extra oxygen around, there's a problem because rubisco, that enzyme in the Calvin cycle, also has an affinity, also can bind to oxygen. Rubisco evolved before oxygen was a real common part of the atmosphere, and because of that, its active site also can stick to oxygen. It prefers CO2, but it can stick to oxygen, and when it does do that, a process occurs known as photorespiration. And this is wasteful. It's not good. It doesn't help the plant. It wastes energy, okay? So a few plants that live in hot environments have evolved ways to avoid photorespiration. One way of doing this is known as C4 photosynthesis. It's known as C4 photosynthesis because one of the first molecules produced in the Calvin cycle in this plant has 4 carbons in it instead of 3 carbons like most plants. So what happens in the case of C4 plants, excuse me, we separate the light reactions from the Calvin cycle into different cells, different places. Why? Because the light reactions, which take place in the mesophyll, that's where CO2 comes in, right? It's actually going to be stored as an acid. Instead of just used directly, it's going to be turned into an acid, a molecule that's bigger and can be transported easily, and it's going to be pumped into the bundle sheath cells. Now the bundle sheath cells, if we drew a leaf again, and I'm not going to draw it in great detail, but if we drew a leaf again, we had mesophyll up here, right? We have mesophyll down here. This is where the light reactions are taking place, right? In these mesophyll cells. The bundle sheath cells are cells that surround the veins of the leaf. They are cells around the vein. And this is where the Calvin cycle takes place in these plants. So it's different than where the light reactions are taking place. So they pump all their CO2 into these cells. Why? That way there's a high concentration of CO2 here. So there's a high concentration of CO2 here, and there's no oxygen in here. All the oxygen is released from is being produced in these cells. So O2 is being released in a different cell instead of in this cell where CO2 is needed. So you don't have a high oxygen concentration down here because these cells are not producing any oxygen. So you have a lot of CO2 down here, and that allows the Calvin cycle to take place. They have a lot of CO2 because it's stored as an acid and it's pumped into this cell and then converted back to CO2 for the Calvin cycle. Now this costs energy, but it does not cost as much energy as fixing photorespiration. Okay, so once again, in this case, we separate the light reactions from the Calvin cycle into different cells. Now in another type of evolutionary solutions known as CAM, for Crassulacean acid metabolism, you see this in pineapple. What they do is they separate the light and dark reactions out in a different way. What they do is they bring carbon dioxide in at night when it's not hot, so they don't dehydrate. They bring it in and they store it once again as an acid, as an organic acid. And then during the day, in fact, they don't actually separate out the light and dark reactions into different cells. They separate when they bring CO2 in and when they release it. So they bring CO2 in at night when it's not hot, and they release it during the day inside the cell and run the Calvin cycle. And that prevents photorespiration. So there's always a lot of CO2 around because they bring it in at night when it's not hot, when they don't dehydrate, they store it, then they release it during the day. So those are two ways that plants have evolved to avoid dehydration and to avoid having more oxygen in their leaves than carbon dioxide. In one case, they put the oxygen releasing part of the reactions in a different cell, and they store CO2 and they push it towards the cells where the Calvin cycle takes place. In another case, they bring CO2 in at night and they store it and they release it during the day. So that way, even if their stomata are shut, they have stored CO2 inside that they can release throughout the day. Pretty cool. And of course, all of this photosynthesis stuff, once again, ties into the global flow of carbon in the atmosphere. Photosynthesis is able to take CO2 out of the air and turn it into a solid. That wood of an oak tree, the wood of an oak tree is made up of old carbon dioxide that has been turned into a solid. So trees are carbon sinks. What that means is they have CO2 from the atmosphere that they pull down and they turn into a solid. And if they die and get buried, that CO2 is removed from the atmosphere. Now, if that tree dies and is burned in a fire, that carbon goes back into the atmosphere again. But plants are a vital part of this cycling of carbon throughout the ecosystem. We've mentioned that energy flows through an ecosystem, but molecules like carbon cycle. And what we need to do as humans is stop releasing too much excessive CO2 into the atmosphere, and we need to get more trees planted and things to pull CO2 out of the atmosphere and then lock it up into solid form. Because what CO2 does is it reflects heat energy back down to the earth and causes the earth to hold on to excessive heat, much like putting a blanket around the planet. And that's where some of our problems with climate change are coming from, is the early stages here of this climate change process. Okay, so that has been photosynthesis. I'm going to post a few things to help you as well as these videos on Blackboard, and I hope you're able to review the video, it makes sense to you, and you can of course email me if you have any questions. Thank you.